Hello and welcome to our content drop today. We will be speaking with Mr. Blankenship, who has over 30 years of experience in developing and implementing biomedical devices. Our discussion today involves his perspective on his biomedical experiences and projects. And with that, I will hand it to Mr. Blankenship.
Thank you, Lauren. I'm Larry Blankenship. I'm pleased to be here today. And as she said, I've done a lot in the biomedical engineering field. And so I will share with you some of the things that I've done, and you can think about whether this is a career path that might be right for you. What is a biomedical engineer? What is biomedical engineering? It is a very difficult thing to define. Here from LinkedIn's endorsements section are two biomedical engineers. This one on the left, you can see, says biomedical engineering and has worked in cell culturing, in biomaterials, microscopy, tissue engineering, very interesting areas. Now, the one on the right has worked in startups and entrepreneurs, product launches, product development, commercialization, worked in hospitals and so forth, but they're both biomedical engineers. And what between the two is common? Only the term biomedical engineering. So if you want to get into this field, you're going to be faced with a lot of really interesting possibilities and decisions. If we take a look at areas of specialization, you can go into research, and that research can be in a corporate environment, a university, a hospital. You can actually be working with patients in clinical trials. If you're looking at developing products, again, it can be in a corporate or institutional environment, or in my case, I've started a few companies. That's entrepreneurial. You can do that as well. Another area of specialization for biomedical engineers is actually helping the people in hospitals with the equipment. Somebody's got to set it up, somebody's got to support it, somebody's got to sell it, somebody's got to work with the hospitals to make sure the equipment is working for the doctors and nurses. So these are all areas. And within any of those, it could be in pharmaceuticals, it could be in biotechnology, such as drug development equipment or gene therapy, or it could be in medical devices that actually provide therapy for patients, or medical devices that are diagnostic to try and find out what the situation is with the patient that needs to be addressed. My specialties were in medical devices and diagnostics. So I'm going to talk from that perspective. These are photographs of some of the devices I have developed over my career. We're going to talk about some of them more specifically. This is an IV set that goes from an IV bottle to the patient, and these are IV pumps that I worked on to pump that fluid into the patient. This is what's known as a suction wand during surgery to clear the area. But this one's very special. You can see that it has a light on it, and it also has a button to wash off the site so you can get better vision. This interesting little guy here is actually only 2 millimeters in diameter, and it goes through an endoscope to clip off just a little tiny piece of tissue to examine that tissue biologically and see what the problem is with it. Over on the left here, this is a catheter. It's called a urological catheter with a balloon that holds it in the bladder. And what we did with this one is we put a temperature sensor in the tip so that it could be used to monitor the patient's temperature during surgery. And this is the controller here for the wand above. This is an intracranial pressure monitor for emergency people to use when their patients and first responders have a head injury. This is another IV pump, and you can see it says kids on it. That we did this one specifically for children's hospitals. And down in the lower left, we're going to be talking about this one in some detail. This is an interesting system that actually takes cancer cells out of patient's bloodstreams. These are very flexible, thin endoscopes that allow you to see inside the body, along with this piece here, which you can see is the instrument that provides the screen. This is a two-part heart valve, and this shows it actually being installed in the heart in a patient. This is a screening device to keep particles from going into the brain. We're going to talk about that one. And this large picture we're going to talk about last, because this is a very interesting device that helps bring people back from sudden cardiac arrest. It's associated with CPR, or cardiopulmonary resuscitation. With that in mind, let's go take a look at a couple. In engineering and in science, we use what's known as the scientific method, which really has these steps. You observe something and you think about it, and then you speculate what might be going on. You then come up with a hypothesis of what you think is happening, and you design tests to see if your hypothesis is correct. Once you've got that, then you can implement something to try and put it to work. Let's give an example here. Cancer tumor cells, also known as CTCs, are not as flexible as blood cells are. And so what we're going to do is to try and filter them out of the blood mechanically. Let's take a look at how that might be done. This is the Viatar oncophoresis prototype system that we built and is in use in testing today. And we're going to talk more about this, but let's let the people at Viatar tell you directly what they're doing.
Viatar, the cancer dialysis company. Cancer touches everyone. It is one of the leading causes of death worldwide. Well before an MRI can detect cancer, millions of cancer cells detach daily from a primary tumor. These cells, known as circulating tumor cells, or CTCs, cross into the bloodstream. CTCs then travel throughout the body, carrying the primary tumor's DNA. CTCs then seed secondary metastatic tumors at distant sites, such as the lungs, brain, bones, and liver. Over 90% of all cancer deaths are due to metastatic tumors, not the primary tumor. The Viatar therapeutic oncophoresis system is an investigational device for patients with metastatic or stage 4 cancer. It is easy to operate by a technician in a clinic or hospital setting. It has none of the adverse side effects of chemotherapy. It will cost much less than new cancer drugs. Viatar's therapy uses an extracorporeal fluid circuit and filter, much like dialysis. The key feature of Viatar's technology is how it separates CTCs from normal blood cells. Normal blood cells are pulled through small conical pores in the Viatar filter at a gentle flow rate, just like in a capillary. But CTCs are larger and less deformable, so they stay behind and are captured. This unique design passes 5 billion normal blood cells per second back to the patient without clogging or causing hemolysis. At the end of the treatment, a substantial majority of CTCs are retained in the filter, and virtually all of the extracorporeal blood is rinsed back to the patient. The tubing set and filter are discarded, while the captured CTCs can be saved for genetic and molecular analysis. Viatar's oncophoresis system processes 10 liters of blood in a 3-hour session. The frequency of treatments depends on how a given patient responds. Some patients may respond favorably within a few weeks because CTC removal triggered an immune system response. Other patients may need long-term treatment, like patients with AIDS or kidney failure. Numerous animal and human studies demonstrate the principle that Reducing CTCs leads to fewer metastatic tumors and longer overall survival. Since 2004, the FDA recognizes that CTCs are a prognostic biomarker for overall survival. Viatar's oncophoresis therapy aims to slow the progression of metastatic disease by reducing CTCs. The goal is to improve overall survival and make metastatic and later-stage cancer a chronic rather than fatal condition.
This was a fun development project. It is in testing right now, but they talk about the conical holes. There were 200 million of them put into a plastic sheet by a laser. That was a fun development. All right, so that's one interesting project. Let's go to another one. We talked about the heart valve. We're going to start the motion in a second, but what you're looking at here is an ultrasound of a beating heart. And when the colors come up, the colors use a system called Doppler measurement, which shows their motion. So we're going to start this, and now you see the heart. Now we're going to turn on the color, and what you see in the color is no particular pattern. It's a mess, and that's because this patient's heart valve was not closing properly. So now we're inside the heart is beating, has stopped, and we're trimming out the tissue that is not working properly, and we're going to put in a new valve. Before we put it in, though, we want to size it. That's a 25 millimeter circular sizer, which shows that it's right for this patient's aortic root. And now we're going to put in the sutures, and we put those little pieces of plastic called plagettes to make sure that the sutures don't tear through the tissue itself. This is the base of the two-part valve, and what they're using there is called a sewing cuff for obvious reasons. They're suturing through it. And what we're going to do after they rinse it in a little bit of antibiotic solution is what we call parachuting it into position, and that's because of all of the sutures that are involved. Looks like a parachute. Now we'll remove the handle by unscrewing it and let the doctor actually suture the base to the tissue. Now we're going to clip off the holder and expose these three thin plastic retainers. They push the tissue out of the way so that when we're ready to put in the leaflet set, it will go in smoothly. So now doctors love sutures, so we do everything with sutures. They clip the sutures and that releases the part. And in this particular case, you can see right through to the ventricle mechanisms of the patient. So if there's any debris, we can take it out. Here's the leaflet set itself. The leaflets are made of bovine or cow tissue and treated to make them very thin, like a leather. We clip them over the three posts that hold it in position. And I'm going to stop this for just a second here after we take off that handle and the holder for the leaflet set. So I'm stopping this. What you have here is a beautiful tri-leaflet valve that will completely replace this patient's previous valve and give them proper valve function. So now, after we clip off anything, we're going to close the heart and restart it. There's the beating heart again. And now let's look at that ultrasound and turn on the Doppler color, and notice it looks like a valve now, on, off, on, off. This was a wonderful product to develop. We were working here with the Hanover Medical School in Hanover, Germany, and these valves worked quite well. Let's go to another example here. It's also in valves because that was a surgical valve implanted by opening the chest. But there's another way to do this, a new way to do it. a transcatheter approach to replacing the aortic valve in the heart. Here is a video talking about how that process is done.
The TAVR procedure involves accessing arteries in the legs and going up the aorta and crossing the aortic valve. The aortic valve is the sick valve in this case. You cross it with a wire and you work over that wire. You dilate the valve with a balloon, and then you place the valve, which is mounted on a stent, through that sick valve and expand the stent.
This delivery catheter allows us to put the stent valve directly on top of your old valve and then sort of inflate it the way we do stents or balloons of that magnitude and allow a new valve to be set in that area. And that's sort of the whole process, is once the delivery catheter gets to the area, inflate the balloon expandable stent, and then the valve is then positioned, and then the catheter comes out and the procedure's over.
So as I say here, this is an important new technology, but the problem with it is it can occasionally cause strokes. In about 2 to 5% or so of the cases, some of the particles that are loosed into the bloodstream when it opens go up to the brain. We're going to look at the anatomy in a minute. So a project that I'm currently working on is to prevent that problem. We're working on a product we call the Point Guard for cerebral embolic protection. It keeps protecting the brain during the procedure from the particles that are generated by this transcatheter TAVR procedure. Let's take a look at this animation, and I will narrate it as we go along. So here's the anatomy. Here's the aortic arch, and these 3 vessels at the top feed the brain. So what we do is we bring in a catheter and open up a filter, and that filter covers all 3 of those vessels to keep any particles that are generated during the procedure from going into the brain and potentially causing a stroke. Here's a fluoroscopy image in an actual human. Now, we have our Point Guard in first, then they go in with the TAVR catheter that's going to put the new valve in place. And when they put that new valve in place, these gold pieces show particles that are generated by that process, but we're filtering them out. We're keeping them away from the brain. Now, after the whole procedure is over, we remove our protective catheter. And so the idea of this is to take that secondary problem of potentially causing strokes and eliminate that to make the procedure safer. Remember the scientific method. We're going to observe, speculate, hypothesize, test, and implement. So let's look at CPR. CPR is cardiopulmonary resuscitation. We've all seen pictures of it where someone is pushing on someone's chest, breathing into them to help them recover from a sudden cardiac arrest. Unfortunately, the data shows that only 1 in 10 or less than 1 in 10 people actually recover from this. And then those who do recover have brain damage. Why is that? Well, it's easy to say that, well, the patient was without oxygen for a while, so their brain damage is from a lack of oxygen. We worked with a doctor in the University of Minnesota who was not convinced that that was the whole story. So he did some tests on cadavers, and he showed that when a patient is lying flat and the heart is stopped, if you hit them in the chest hard enough to try and restart the heart, it puts a hydraulic pressure waveform to the brain, and it's so high in pressure that it's like you hit them in the head with a baseball bat. It's a concussion. And a typical CPR procedure has between 1,500 and 2,500 compressions. That's 1,500 to 2,500 concussions in each time you try and do this procedure. So how do we deal with this? Okay, First of all, let's look at what the first responders use today. Whether you're doing mechanical or manual CPR, as we talked about, pushing on a patient's chest, or using a new mechanism called the Lucas, doesn't matter. The survival rate's only 6 to 10%. But the Lucas is carried by most of the first responders. Let's take a look at it here.
Siapa jaga adrenalin? Every 3 minutes.
So by putting this Lucas on a patient, that does the pounding of the chest, and it allows the first responders then to deal with other things such as the airway, putting an IV line in the patient, or whatever else is needed. And if you did not understand what they were saying, it's because it was in Swedish. The Lucas stands for Lund University Cardiac Assist System, and Lund University is, of course, in Sweden. Now, how do we deal with this? Well, our doctor in Minneapolis came up with an idea. Why don't we use gravity? We're going to hypothesize that if we raise the head, the gravity will help counteract that compressive force, and maybe we can do our job and not have the damage to the brain and actually recover more patients. So we started by working with animals, and we used pigs in this case, put them into the same condition that a human would be for recovery. And we found that by raising the head, as you're shown here, we can actually increase the recovery from 10% to over 40%. That's significant. That means that we could actually increase the number of patients that we recover by 4 times. Recover these patients back to life. This is amazing. But we also found that if you just put a wedge under the patient and then try to start the heart, it doesn't work, because then you're working against gravity. So the trick to make it work is to start it while the patient is essentially flat, and then move the head up very slowly. So if you've got the pump, the heart primed. All right, this is the device. I had the privilege of leading the design team on this, and the company is calling it the Elegard. This is the Lucas backplate. So we incorporated that right in the design. So when the patient lays down on it, they can snap that Lucas mechanical device right over them to keep the heart pumping. It's shown now in the up position. This is a soft rubber pad for the neck, and the patient's head rests in this cradle here. And then this is the control panel. So when it's in the down position, the patient is essentially flat, although we've got the head up just a little bit. And you can start the CPR and then push the button, and it will raise the head up to the up position in about 2 minutes. This product was developed over a few years and then introduced into commerce in the summer, early summer of 2019. Shortly after that, it was used in a case at the Minneapolis St. Paul International Airport, and this television news account of that case will tell you a lot about this career. This shows you why we do what we do. And I will tell you, I have found it a very satisfying and interesting career, and I'm glad to be doing something to help people. Let's watch the video.
Heart attack, but is alive tonight thanks to a quick response by a TSA agent.
And Fox 9's Iris Perez joining us live from the airport tonight. And Iris, you spoke with the passenger and his wife. This was a pretty scary situation.
Most definitely it is, Kelsey, and first responders had to shock Greg Eubanks 8 times before a heartbeat returned. And had it not been for how quickly TSA agents jumped in to help, Greg says he wouldn't be alive.
I look at August 10th, and that is basically my new birthday now.
From their Southern California home,
I was given a second chance, and I'm just so thankful.
Greg Eubanks and his wife Laura celebrate his new lease on life.
I just looked at him while he slept after I got him home, and I just cried. I just couldn't stop crying. I was so thankful. So relieved.
It was August 10th. After visiting his parents in Indiana, Greg headed back home. His connecting flight to San Diego at MSP, where, without warning,
I just collapsed to my knees and fell face forward.
Fortunately, 2 TSA agents, Brittany Sutton and Eric Jones, rushed to help.
They were able to respond within 30 seconds of that.
He got the entire bundle of care for cardiac arrest resuscitation,
Including with Elegard, a new device that allows for head-up CPR.
The airport is one of the first places in the nation to have this device. They just implemented it.
At the hospital, Greg, a father of 4 with 1 grandchild and 2 on the way, was in a medically induced coma while his family scrambled to Minneapolis.
If it weren't for how quickly these people acted, Greg would be dead.
Once Greg emerged from his coma, the family began an online search for the agents who saved his life. Later, they even stopped by to visit.
When I first met them, all I could do was just hug them and just cry.
They're heroes.
Why the Eubanks plan to keep these lifesavers close,
They are a part of my extended family right now.
even from afar.
I just want them to know that I love them with all my heart, and I owe everything to them.
Now, Greg says 5 days after he woke up from his coma, he was cleared to go back home to California. He's taking things one day at a time, and that if Brittany and Eric don't go to San Diego to visit him, he'll make sure he's back in Minneapolis to visit them. Live at the airport, I'm Iris Perez, Fox 9.
So this is one example of many of why we do what we do in biomedical engineering.
Heart attack, but is a
Thank you so much, everybody, for listening to Mr. Blankenship's discussion. If you have any follow-up questions for Mr. Blankenship, you can follow his LinkedIn, which is on this page here, or email him directly at larry@blankenshipresearch.com. And of course, if you want to participate in any future events or see some other content from the Bioengineering Empowerment Program, definitely check out our website, engineering.ucdenver.edu/beep, or email us at beep@ucdenver.edu. So thank you all again. Have a wonderful rest of your day.